Welcome to Chapter 11, the basic principles of heredity. Some of this will be a bit of a review, but for sure some of it will be new for all of you. So, Gregor Mendel was sort of the father of sort of modern genetics. It's not really that modern anymore, but he was the one who initially deduced patterns of inheritance, or at least the simpler patterns of inheritance. So, take a look at one example of some of his initial observations. Mendel crossed some yellow peas with some yellow peas. Most offspring were yellow, but some were green. So if you're a scientist and you're studying this, what would you think? You'd think, I have no idea what this means. Perhaps it is simply random. Perhaps something in the environment caused them to be green. Mendel started collecting a tremendous amount of data and eventually was able to put together a pretty good idea of what was going on. So we'll come back to this a little bit later. Some of you might be able to already answer this. What we know now that Gregor Mendel did not know then. We know about chromosomes, genes, and alleles. This is a reminder to you from our last chapter on meiosis and mitosis of what a chromosome is. We know that chromosomes are made of DNA and protein. They have genes. Genes are heritable factors that control specific characteristics. And then we also have alleles, or different forms of a gene. So this might come back to that last slide. Green would be one allele. Yellow would be another allele, and these are of the same gene. So somehow these parents that were all yellow gave birth, whatever, I mean they're peas, whatever they do, gave birth to a whole bunch of yellow and also some green. We now know also that on these chromosomes there are what we call gene loci, or a gene locus for singular, specific position of a gene on a chromosome. And a review from maybe a little longer ago, all individuals of a species carry the same genes at the same loci on the same chromosomes. This one I really love to, okay, I don't love to, I mean that'll make me sound mean, but I'm always kind of interested to ask the class this question at the beginning of the course, and even after we've learned it. I ask them, does everyone in this room have 100% the same genes? And basically everybody says, no, we obviously don't have the same genes. I mean, we're all different people, right? But of course we do have the same genes with some differences between men and women. But other than that, we have 100% the same genes, but we have different alleles, so different forms of the same genes. If we didn't have the same genes, that would mean that whoever it is that had these other genes, they are a different species. Some definitions that we need to go over, and some of this will be review. I recommend that you do this. It's a great reference for you to look at when you're reviewing for your exam. So you can copy it into your journal or notebook. Next, what we're going to do is label it with the following terms: centromere, allele, carrier, all these terms. We're going to label it. Some of them you could probably label already, and some of them are a little bit interchangeable. Let's see if we can figure out which ones are. So definitions. This image shows a pair of sister chromatids. Did you catch me what I said that was wrong there? They're not sister chromatids. How do you know? Because in the middle, the centromere is not connecting them. Also, we've got kind of some different symbols down here. They're not sister chromatids. They are homologous chromosomes. So here we go. Name and annotate the following labels. Okay. Oh, I forgot. Let me just restate what this is. This is a karyotype, all the chromosomes of one individual. And what we see here, this whole thing would be one of these. And if this was shown, let's say during metaphase 1 of meiosis, then we would see those X structures that are connected in the middle. Those are the centromeres that are connecting them. Here, the centromeres are present, but they're not connected. So these are not sister chromatids. Here are our labels. Let's start with the top. So what is a genotype? This one could be labeling almost, well, actually all of these. A genotype is the combinations of alleles of a gene carried by an organism. So this one right here, we would say their genotype is AA. This one right here, we would say their genotype is little b. And let's go down to one that's different. The genotype for the individual here is big D, little d. Some of this is probably starting to remind you of high school now. Okay, so phrases, terms to know. What's the next one? Let's go to phenotype. I know we're not labeling them yet. You're all probably hating me now. The characteristics often resulting from an interaction between genotype and environment. So eye color, for example. What is your eye color? If it's blue, we'd say your phenotype is blue. And your genotype, well, we might not know what your genotype is. So phenotype is what the observable trait. Observable trait. Centromere, we already know that one, joins chromatids at cell division. Alleles are the different versions of a gene. Now, two new terms here. Reminder from high school, though, probably. Dominant alleles are expressed even when the other alleles are present. So for this individual, usually we represent a dominant allele with a capital letter, at least for instructional purposes. When you get into scientific literature, there are all kinds of crazy abbreviations. It would be like H delta S star Q or something like that. But thankfully, we don't do that. What we do is just a capital D. That means it's dominant. It even stands for dominant. And then this little d, that's recessive. And if this big D maybe stood for dark eyes, and that's the allele for dark eyes, and this one's the recessive allele for light colored eyes, and this person's big D, little d, then they would have dark colored eyes because the dominant alleles are expressed even when other alleles are present. So this one is basically ignored, and we see this one, the big D, becomes the phenotype or observable trait. Big D, I said, coded for dark eyes in this case, so okay, this person's phenotype will be dark eyes. All right, recessive alleles, these are only expressed if the individual is heterozygous, and, excuse me, what, did I say that? Homozygous. Up here, this is an example of homozygous. This is also an example of homozygous. In this case, so lowercase, that means that this individual is homozygous recessive, has two copies of the same recessive allele, and they're only expressed when both alleles are recessive. So the classic example, which is in fact not true, if little b and little b, each one of these stands for blue-eyed allele. So from mom, got a blue-eyed allele. From dad, got a blue-eyed allele. All right, you've got blue eyes. If that's a fun example, and it's like appears to be true, but it isn't quite true because there's other genes involved. But anyway, up here, this is a homozygous dominant individual, and this individual is going to have two copies of the same dominant allele. Capital letter, again, generally means dominant. Codominant. So what I've talked about so far, students do not make a lot of mistakes on those, except for alleles. They'll often confuse alleles with genes. Dominant and recessive is pretty simple, though. We're going to go back over that in much greater detail. But this one, notice the notation is a little bit different. You do need to pay attention to that. So I'll draw it a little bigger. So there's a C, and then there's something else, a superscript up there. You do need to write these things that way, and we're going to study codominant later. These are pairs of alleles which are both expressed when present. Use superscript. So both expressed, let's see, I'm going to give you a fake example because all the real examples are a long story. A fake example would be you have the brown-eyed allele and you have the blue-eyed allele. And if they were codominant, then we'd write them differently. We'd write C, big B, and then C, little b. And somehow, magically, you're expressing both the brown eyes and the blue eyes. So when somebody looks deep into your eyes, they notice, you kind of got both, brown and blue. Next one is heterozygous, and that's having two different alleles for the same gene. This last one down here, this one also we could call the same. This is also heterozygous, but sometimes we'll use another term. We will say that the individual is a carrier, and this a student mentioned this actually earlier in the discussion regarding not sickle cell anemia, but cystic fibrosis. Carrier is a carrier. That means heterozygous, and they're heterozygous for a recessive disease-causing allele. So that would mean essentially the little e codes for some type of disease, but it's recessive. So this individual doesn't have the disease because they've got one dominant copy that codes for no disease. It does mean that it's possible their offspring might inherit this disease-causing allele, and it could cause some problems potentially. Okay, and then gene loci, plural for the location of specific genes on a chromosome. All right, so this is again homologous chromosomes, not sister chromatids. They have different alleles, and they're not joined at the centromere. So we're going to now look at an example of the types of experiments that Gregor Mendel did. Usually when you look at genetics, you are going to often have kind of a key to phenotypes and alleles. So phenotypes are what you can see, so red or white, and then the alleles are something like letters or numbers or something. So giving a code is a sure way to make sure you're being clear. So the capital R means red and the lowercase r means white. And what we've learned is this capital R means that allele is dominant to the white allele. You need to know this term. F sub 0, it's also referred to as the P generation, otherwise known also as the parent generation. And in typical genetics crosses, what we're interested in is finding pure breeding individuals, and then we breed them with other pure breeding individuals. What pure breeding means is that they are homozygous for a given trait. We can also say pure breeding because before we knew all this homozygous terminology and whatnot, we could just say that all their offspring, when they're bred with another thing like them, are the same. So pure breeding white, if you breed a pure breeding white with another white flower, that's not what's done here. But if you did do that, white with white, then all the offspring are going to be white no matter how many times we do this. This flower has 1,000 offspring, they're all white. So we say, wow, very pure breeding for sure. So we've got these pure breeding, this pure breeding parent generation of two different phenotypes, different, red and white. The F1, the observation is this, they are 100% red. F stands for filial, I'm going to write it out, filial, one meaning the first generation after this pure breeding parent generation. Then we do a self-cross, which means flowers, it's okay for them to reproduce with their brothers or sisters, or even themselves. Yes, they're special. They can often reproduce with themselves. So we self-cross them. So whatever this offspring was, we breed it with itself. That means if any of you are into plants, plants can self-pollinate. Some of them can. So we breed it with itself. Oops, I went a little too far. Anyway, the result in the F2, or second offspring generation, is 75% red and 25% white. And if we were to repeat this, we're always going to get about these numbers. We won't always get exactly 75%. So, for example, if you had out of 1,000, you'd probably get something like maybe 780 red, and then what would it be, 220 white. But roughly this ratio, sort of a 3-to-1 ratio every time when we do this experiment over and over and over. 75% to 25%, or 3 to 1. What we now know is, well, and also what Gregor Mendel came up with, is this simple system of there seems to be 2 copies of every gene in these individuals. And he also came up with the idea of dominant and recessive. So he decided that white, I'm going to say that's recessive to red, because when you cross a pure breeding red with a pure breeding white, you get 100% red. He also came up with something called a Punnett square to organize his thinking and show how mathematically how he believes this works. He believed that there is two copies of every gene, and when it's pure breeding, it's not very interesting, but basically you could, a gamete or sex cell, in this case it would be, if it was the male gamete, it would be pollen, and if it was the female, it would be ova or egg cell, ova or egg cell. So in this case, same difference. So you could get this little r on the right or this little r on the left, right? I got my left and rights wrong. Same thing over here. From this parent, you could get the big R on the right, or you could get the big R on the left. These represent gametes. I'll come back to that a little later when I show that they can be different. The insides of the squares represent the fertilization. So this is, well, maybe we'll say after fertilization. These are possible offspring genotypes. Possible offspring genotypes. In this case, there's only one possibility, and it's repeated 4 times. Each of them is heterozygous, and they're all red because red is dominant to white. When we do the self-cross, we are going to draw another Punnett square, but this time, because the parents, so the parent, we're going to do a self-cross, so big R, little r, crossed with big R, little r. Again, with each parent, 50-50 chance. If a gamete gets a little r or a big R, this parent, same thing, 50% big R and, whoopsie, 50% little r in the gametes. It's random which gamete is going to fertilize another gamete. Could be that the offspring gets a dominant allele for red from both parents. Could get one of each, could get one of each again, and could get both of the small recessive alleles for white color. Now, notice that there are 2 ways that we could get a heterozygote. It could be that the offspring, so this is the possible offspring genotype, right? Could be the offspring receives the recessive allele from this parent. It could also be that they receive the recessive allele from this parent. So there's just 2 rearrangements, basically. Result is this. 25% are homozygous dominant, 50% are heterozygous, 25% are homozygous recessive, and 3 out of 4 are red. So all these are red. Notice 25% plus 50% equals 75%. And notice that 25% plus nothing equals 25%. And in case you wanted to review those terms already, I left them here for you. Homozygous, remember what those are? Slightly different definition, but same idea. Heterozygous, F1 generation, offspring that results from a cross between an organism with a homozygous dominant genotype and one with a homozygous recessive phenotype. That is the definition of that. You do need to know it. And then the F2 generation, the offspring resulting from a cross between 2 F1 heterozygous organisms. It could also be a self-cross, but that's only with plants, which can fertilize themselves. Animals cannot do that. Like if you've ever had a pet cat and it got pregnant, it did not impregnate itself, all right? So you were thinking that, now you know better. So what Mendel did was he performed crosses such as the one I just discussed with many traits. You can look up, there's a table in the book with lots of different traits. He did the same type of analysis with the parent generation, F1, and then F2, and he saw the same pattern emerging, that there'd be something like maybe tall crossed with short, and then the F1 is all one phenotype, and then the F2 is a 3-to-1 ratio. He came up with this idea, the law of segregation, and that is this: that these 2 copies of the sort of trait, the yellow and the green copy, basically, they segregate and produce sex cells, such as pollen. So taking his yellow and green example, so capital Y, that's yellow. And then a little y, that would be green. This is after DNA replications happen, so there's the sister chromatids. They're identical and they are attached to each other. But basically, what happens, we know now, is that the alleles are going to segregate. So half of the gametes, this half here is on the right, is only going to get the little y version. We're going to ignore the crossing over for now, okay? Just forget about that. And then the one on the right is going to get the dominant version. So, and then meiosis, we get another division, right, to get to divide the sister chromatids. And so this chromosome has got a capital Y, this one's got a capital Y, and then these two would have both lowercase y's on them. And so these gametes here will both go to, if they were to fertilize, be fertilized and become a zygote, they would contribute a dominant allele, and the ones on the right would contribute a recessive allele. So we can now explain this in much greater detail. The yellow parent peas must be heterozygous, right? So big Y, little y, big Y, little y. The yellow phenotype is expressed in both of the parents. Through meiosis and fertilization, some offspring peas are homozygous recessive. They express a green color. So both of these parents, remember segregation, we're in meiosis, we're going to produce haploid sex cells or gametes, and 50% of the time, one of each is produced. So we take this parent first, big Y is going to be inherited half the time, little y the other half. Now if we take this parent over here, big Y half the time, little y the other half, and then we fill it in, big Y, this is homozygous dominant, big Y right from here, and then little y, and then big Y right from here, and then little y from here, and then little y, little y. All right. So this individual right here is the green one, and he did this with huge data sets. He had something like, like I said, on the order of 1,000 offspring, because peas, if you've ever harvested peas or grown peas, they have a lot of offspring. Each pea is an offspring, so there would have been something like, maybe 720 here, and then here there was 280, roughly a 3-to-1 ratio. And as we see over here, it is 1, 2, 3 ways we can produce a yellow pea, and 1 way we can produce a green pea. 3-to-1. So you can see that. So just what I already said, Mendel didn't know about DNA, chromosomes, meiosis, none of that. Through his experiments, he did work out that there are these heritable factors. They're passed on from parent to offspring, and that these can have different versions, and that there must be 2 copies of each gene. And he worked that out based on math. So again here, this is basically the same thing that I just explained. I'm going to add a couple more details here. So remember, alleles segregate. That's what's going on right here. Segregation of alleles, big Y or little y, big Y or a little y. That happens in anaphase 1 of meiosis. And we can use a Punnett grid square like the one shown here. I realize I kind of covered this one in the last slide, so moving a little faster. Genotypes, these are the genotypes down here. We can have homozygous dominant, heterozygous, or homozygous recessive. And there's another thing here that is important to know, and that is this can often be called, not often, but it is called the phenotypic ratio. So the ratio of traits that you can see. 3 yellows for every 1 green. This is an important one for you to know. So monohybrid crosses. Monohybrid crosses are what refer to crosses that involve 1 gene with 2 different alleles. 1 gene, 2 alleles. Sometimes it's different. It could be like a trait that is controlled by many genes and maybe many alleles too. So we're going to go through another example. Example question number 1 of 3. So what happens if we cross a homozygous recessive with a homozygous recessive? What are the genotypes going to be, the phenotypes, and the phenotypic ratio? You can pause it here if you'd like to think about it, and then I will show you the answer. There's the answer. Homozygous recessive crossed with homozygous recessive. Genotypes are all little y, little y, little y, little y. So there's only one possibility. They are all green. Example question number 2. Homozygous recessive crossed with a heterozygous individual. I'll pause it, see if you can fill out what are the genotypes going to be, the phenotypes, and the phenotypic ratios. Or if you really want to go through it a little more slowly, you can fill out in all of those as well. Okay, I'm going to show you the answer now. It is as follows. So this heterozygous individual half the time gives a recessive allele, half the time gives a dominant allele. If one of the parents gives a dominant allele, then the color will be the dominant phenotype, the yellow phenotype. So all these are yellow here. And if they give the recessive allele, then they'll be green, always. Why always? Because its partner, or P, only has recessive alleles. So it's either recessive or recessive. It's not really a choice. It's always recessive. Example question number 3. Homozygous dominant crossed with heterozygous. What would the genotypes be? Phenotypes, phenotypic ratio. Here we go. They're all yellow. The only thing that is different is that half of them, half the offspring, are heterozygous. Big Y, little y, big Y, little y. You can look at the diagram a little more if you want. I'm going to move on. Test crosses. What a test cross is used for is as follows. Used to determine the genotype of an unknown individual. The unknown is crossed with a known homozygous recessive. So you can ask yourself, why is it always with a homozygous recessive? Well, a homozygous recessive, firstly, only can produce gametes that have the recessive allele, and why not use homozygous dominant? Why not use homozygous dominant? Well, let's go through the example here. Possible outcomes are this. So we have an unknown parent, and that's the unknown parent would be red, right? Because that's always their phenotype, and we're wondering, are they big R, little r, or are they big R, big R? If all of the offspring are red, what we know is the recessive allele certainly came from the homozygous recessive parent that is in the test cross, obviously came from there. And if all of the offspring are red, then we know that the parent must be homozygous dominant. You would need to repeat this many times because it's certainly possible that if you only did it, say, 2 times and they were both red, that it was just kind of luck of the draw, and the parent, who is heterozygous, just both times, gave the dominant allele. So the other possibility is this, that the offspring, some of them are white and some of them are red. So this unknown parent in this case is going to be heterozygous. Again, here are the gametes from the test cross individual, test cross plant, homozygous recessive. And if we have any white offspring, then what it means is that the unknown must have been homozygous recessive, gave a big R and a little r. Half the time big R, half the time little r. So half of the time, the result was a recessive phenotype. The next topic is codominance, and codominance is where alleles are codominant, they are both expressed. So it is not where the dominant allele covers up the recessive allele, such as the topics, the traits we were just discussing. It is where they are both expressed. So the name codominance makes sense. Both alleles are expressed. Next, we're also going to look at 2 things here. So the first thing, 1 is codominance, 2 is this. Some genes have more than 2 alleles, so more than 2. Now, what that means is that, remember, a single individual only is diploid, and so they just have 2 copies of each gene, one here, one there. For example, maybe this is a dominant A, and then this is a little recessive one. But for a whole population, maybe this other person for the same chromosome, they also maybe have a big A here, and then maybe their other allele here is maybe uh, A1 or something, so a second version of it. And then just to give you one last example, maybe this person's got a maybe capital A1, and then this one's a little A2. So now we've got, let's see, 1, 2, 3, 4, 5 alleles, and you can have many, lots of alleles. So it's not always this situation where we just have it's either one or the other. So we're going to look at an example of a couple things: codominance and multiple alleles. So this is about the human ABO blood typing, and it's an example of 2 things: multiple alleles and codominance. It codes the gene, that is. Remember, we're just looking at one gene, 3 alleles. The gene codes for cell surface antigens. These are involved in self-recognition and the immune system. We don't need to get too much into them, but basically they code for a protein that is in the cell membranes of red blood cells. So let's go down here. Firstly, there is one allele that determines are antigens going to be present. So this allele codes for no. No protein is present in the cell membrane. And if the person is homozygous, then they're going to have no antigens or no proteins in the cell membrane. So sorry, I got a little bit ahead of myself. Over here, let me just go over the key to your alleles. Key to the alleles is this little I, and this is the alleles I've made up for this. All books have different versions. Little I, we're going to say, is no antigens are produced by that allele. Big I, A, superscript, codominance always has a superscript, remember. That means that type A antigens are present, so this person's at least going to be type A blood. And then the third allele is capital I and then B, which is type B antigens are present. So if a person has 2 of these little i's, that means they're homozygous, and you can see that right here. That means that no antigens are produced in the membrane of red blood cells. If the person has any other alleles, so either the IA or IB, then they're going to have antigen, but it's going to be a question of which one. So down here, the first and most easy example is IAIA. So they are homozygous for IAIA. That means that they are blood type A for sure. There's another option, and that is as follows. They could be IA little I. So let's look at, I gotta resend this. Let's look back at our key to alleles. If they are IA and little I, that means that this one is essentially sort of like it is recessive. It doesn't do anything, nothing shows up. It makes no contribution really to the phenotype in this case. It is essentially covered by the more dominant IA allele right here. Type B is basically the same thing all over again, but it's type B. So the person could be homozygous, have 2 IB alleles, or they could be heterozygous. They'd have IB and then little I, that allele. So in this case, they are heterozygous, and in this case, they are homozygous. Either way, they are blood type B. Last one, and this only in, this one is heterozygous. Heterozygote individuals, heterozygotes, they are what really exemplifies the codominant phenotype and the codominant interaction of the 2 alleles, because they have blood type A and B. We say they're type AB, And that is because they have one copy of each allele. They have IA and IB, so they have A antigens on the surface of the red blood cells, and they also have B antigens on the surface of the red blood cells. So this is heterozygous and is the best way to exemplify what codominance means. And that is all. Incomplete dominance, let me just say this once, even though it sounds really simple now, it is not codominance. It's not codominance. Really easy to get them confused. So here it is. In heterozygotes, an intermediate phenotype is produced. An intermediate phenotype. Both of them are not expressed. So the parent generation here, P generation, red, white, and remember that if it's the parent generation, it's always going to be homozygous and homozygous, or pure breds, true breeding. We mix them, and the genotype is as shown down here, and the phenotype is an intermediate between the two alleles. So it isn't white. Pink is not white. Pink is not red either. It is an intermediate between the two. So when you have codominance, this is codominance. All right. So IB, IA, this is not a mixed, does not produce a mixed phenotype. It produces both phenotypes. The person is type A and B blood. It's not like they're type, I don't know what letter you could put in the middle there. There'd be some intermediate. They're both, right? Both, not an intermediate. So incomplete dominance is intermediate, and codominance is both of them are expressed. So to complete this Punnett square, F1, F2, and all that, if you do a self-cross here between the heterozygotes, you again get that 3-to-1 ratio. Sorry, I'm getting a little ahead of myself. I just realized this lecture is a little bit long. You get a 1 to 2 to 1 ratio. We have a 1 that is homozygous, R1R1, and then 1, 2 that are heterozygous, 1 that is homozygous recessive. Okay, phenotypes are all different. Phenotypic ratio is 1 to 2 to 1. Sex determination. So firstly, I'm going to go over a little of how do we know what determines whether a newborn child in Homo sapiens is a male or female, and also sex determinant species other than Homo sapiens is not required for this topic. Firstly, these are sex chromosomes. We can have 2 of these, in which case the person is a female. We can have XY, in which case this person is a male. The Y chromosome determines sex. So if there is one Y chromosome, then that means it will be male. And why do I say that? You might say equally, well, isn't it just 2 X chromosomes means that the person is going to be female? 2 X's determine the sex. There are some unusual situations where a person may be XX, Y. And in this case, so you have 2 X's and you have a Y, and the person is biologically male, and there are some other symptoms that exist with this because it's an unequal distribution of chromosomes. Now, C is going to reset. Okay, there we go. Now, to show the cross between a male and a female. So, male crossed with female. Actually, sorry, let's get those lined up. Say female crossed with male. Female is XX, male is XY. We'll do it the slow way. Draw the Punnett square out. We are going to show that 50% of the offspring we would expect to be female, 50% we'd expect to be male. The male first is going to, half the time, produce a gamete that has a Y in it. Half the time, a gamete that has an X. The female, all the gametes will be X. So it's either that X or that X. The only difference would be it's all the same genes, right? But there would be some different alleles. Now we fill in the circles, bringing them down, right? Let's bring the X down. Okay, I don't bring this one over there, X, and then this one over there, there, and then this one right over here, and then the Y and Y. So you just count them up. How many are XX and how many are XY? These are both XX. This is XX, and then here is XY and XY. So we have a phenotypic ratio of 1 to 1, also a genotypic ratio of 1 to 1. Dosage compensation. Basically this. In females, human females, one X chromosome in the female somatic cells is deactivated, becomes a bar body. Somatic cells means out and other than the ovaries. All of those cells, they're going to take one of the X chromosomes and make it a bar body. And the reason for that is this. These big X chromosomes, not all of them are associated with managing the sex of the individual. And females have 2 of them. Males have 1. So for all the traits, say, pertaining to how many of some protein is inserted into the cell membrane, we really need there to be not a difference between males and females, because males and females are largely the same at a genetic and cellular level. There's only a couple of differences, really. So what happens here is this one of the X chromosomes is condensed into a small, more condensed version of itself, which is called a bar body, and the genes on it are mostly not expressed, so they will mostly not be used. Next, which of these X chromosomes is deactivated? Well, very early in embryonic development, we've got maybe, I don't know exactly what stage it occurs. We'll say this many cells, right? So which chromosome is deactivated is random. We'll say let's color code them here. There we go. So we'll say the one on the left is blue, and we'll say all these cells had their blue chromosome deactivated. So it should be about 50% of them. About 50% there. 1, 2, 3, 4, 5. Okay. And on the all these other cells, they would have had this X chromosome deactivated. All these ones deactivated. So that what that means is, let's remember that many of the alleles here, trying to get the right color, the alleles here are different. So this one could be, let's say, a dominant allele. I'll say big A. This is the sister chromatid, so big A. And then this one, we'll say on this chromosome, there was a little a there. This chromosome, little a. So what the result is that all these yellow cells are going to be expressing the recessive A allele, and all of the blue cells will be expressing the dominant A allele. And so one of these in cats, not humans, most mammals are this way. In cats, one allele codes for black fur, another allele codes for brown fur, and then there's a different gene actually that is involved that determines the whiteness of the fur. Okay. Oh, and you can ignore this part of it. We're not going to get into that much detail. This is involving RNA, which we haven't talked about enough. Sex linkage. So many of you have probably heard of sex-linked diseases. These are diseases that typically affect males more than females. And what they are is essentially genes that occur in the non-homologous region of the X chromosome, not on the Y. Shown here, this is the Y chromosome. There's the Y. And then this whole thing is the X chromosome. Down here, note we have another non-homologous region. And so basically, this is what goes on. Any gene in the non-homologous region, a male only has one copy of it, just one copy. So because there's only one copy, that means that if it is a disease-causing recessive allele, then the male will be affected. So recessive allele, and it causes for a disease, then males would be affected if they have one copy only. Two examples that we'll talk about of sex-linked disorders are hemophilia and color blindness. Both of them are recessive. Let's see. Last thing to just mention about the X and Y chromosomes. The Y chromosome only has about 180 genes. That's much fewer. How much was it? It was something like, yeah, all right, I wrote it up here. So around 800 more genes on the X chromosome. So females literally, we have all the genes that are in a female, but for these, about 800 in the non-homologous regions, something like that, around 800. We only have one copy. Females have two. So if they get one bad one, they might be helped out by getting a second one from their second X chromosome that is a good one. Okay, one thing that is unique to the Y chromosome, females don't have this gene. It is the SRY gene, and it's a transcription factor. That means when it's produced, it goes around the cell nucleus and activates all these other genes on other chromosomes, causing maleness. So there's a gene on the Y chromosome causes maleness. That's why if a person for some reason is XXY, this Y gene on the Y chromosome is going to cause maleness. If you would like to read about this, it's kind of an interesting little anecdotes from another textbook. This is not actually on your test, but basically it shows that Genghis Khan, what is it, something like 10% of males in much of Asia have the same exact Y chromosome, and the thought is that it is from Genghis Khan because he did a lot of procreating. He himself, we don't know how many offspring he had, but of course all of his offspring had offspring, and well, this Y chromosome was very successful. The Y chromosome remains relatively unchanged because no crossing over occurs. So it is also a way that you can trace male ancestry. So if someone was unsure of who their father was, for example, take a look at the Y chromosome. Each of our fathers will have 100% the same Y chromosome as their son. For females, there's another way, and that's looking at mitochondrial DNA, but that's another topic. Sex linkage. What number do you see here? Well, this has never been super effective here, but I feel like it's a way to make people walk 5 seconds in the shoes of somebody who is colorblind. You see a 2 or a 5? Well, if you saw a 5, then I guess you have normal color vision. 2, then you have red-green color blindness. This is not a real test because I have had students in my class who have told me they are colorblind and it doesn't work. Got this from a site that professed to be a real test, but it appeared to not translate well onto the screen, perhaps. So 5 normal, 2 red-green color blindness. Where is this gene? Well, you should be able to know now. It's somewhere here. It is somewhere here. So we could say that there is maybe a little recessive allele here, and if it is, then this individual will be color blind because the Y chromosome does not have a copy of that gene. If it was a female, I'll draw a couple big fat allele, fat X chromosomes here. Well, they'd have maybe they have the color blindness allele, so that's the coding for color blindness, just like that one. But their second X chromosome, they may have a dominant allele. Dominant, so it's expressed, they can see color normally. Apparently, this is how colorblind people see the world. Again, I'm somewhat doubtful if that is actually true, and I've been yet unable to verify it with any of my students. Perhaps if any of you, I'm certainly not asking for you to tell the class that you're colorblind, but if you felt like it and you are, and you were not able to see what was it, 5, then you might let me know in an email or on the discussion board. So I guess we couldn't see those berries if we're colorblind and stop certain traffic lights could be a little challenging. Okay. So, sex linkage. Sex linkage is another one where we are having to do superscripts. So, the notation for sex-linked genes has got to be like so. Female is always going to be two X's, and then there's a superscript as well. So, if it was for color blindness, you can make up any allele. Here, I'm using an N down here. Let's see, there's N's. It could be C. It could be really anything as long as you provide a key to your alleles when you're discussing what you're talking about. Okay, so female, and then we're going to have some letter up here, maybe a capital P, and then over this one it's a lowercase p. Males are always like so. Remember, and then they will also have maybe the dominant allele there. Remember that codominance is also written with a superscript, but not with a Y and not with X's. All right, so you just choose some letter here. Maybe it could be B. That's okay, and then you write a little T there, and then maybe then the big B again, and then for then a capital T maybe there. Codominant. All right. Next. Where are we? Okay, so next, color blindness. Let's go through the genotypes. X big N, normal female. This is an affected female, pretty rare. That's rare because this allele is also very rare. Something, I don't know what the frequency is, maybe 1 in 500 or something. So if a female gets one of these recessive alleles, the chances that they'll get a second one is also 1 in 500. So really rare. Carrier female, as I said, heterozygotes who have a disease-causing allele are often called carriers. Here, affected male. Affected males only need one copy of the recessive allele, and this is a normal male. Drawing across here, just giving you an example. Genotype for the mom is XNX_n, carrier female, color seeing normal male. And that's how we draw the Punnett square. Hopefully that didn't confuse it. We draw the complete build. There we go. All right, see? So take the female's allele, one there, then bring them across. X big N, X big N. Same thing here, X little n, it goes there, and then X little n, it goes there. And you might be wondering, what about this box here? Should we have written X little n first? Because, I mean, I wrote it first here. By the standard way for genotype notation is the dominant allele first. So the male's allele, not because it's male, but just because the allele is dominant, is written first there. So dominant. And then lastly, the Y is inherited in 50% of the zygote. Lastly, just the phenotypic ratio down here. We can see that there's a 1 in 4 chance of having an affected child in this cross. A couple of example questions for you. Also, I just paused as I was moving on to the next slide, which was hemophilia. And actually, we're going to go over that in chapter 16 in much greater detail, so we'll come back to that later. Mendel's law of independent assortment. We're going to come back to, not come back to it. It's very related to segregation of alleles, though. So what it is the presence of an allele of one gene in a gamete has no influence over which allele of another gene is present. However, this is only true for unlinked genes, which are genes on different chromosomes. So for example, if you have had a child and the child has, you know that it has inherited 2 recessive alleles for gene A, that has no connection to what alleles it will inherit from another different gene on a different chromosome because they are segregated independently of one another. They're not the chromosomes don't move to opposite ends of the cell in meiosis in any organized fashion with regard to alleles. Okay, so this is a bit more complicated now. Dihybrid crosses. Now we're going to be looking at two pairs of homologous chromosomes. Two pairs of H chromosomes. This is not a sister chromatid, right? They're not joined at the centromere, so gotta keep that straight in your head. So one of the homologous pairs has these two alleles for gene Y, a dominant Y and a recessive Y, and the other one has a capital S and a lowercase s, so one's dominant, one's recessive. Key to the alleles is right over here. So the dominant phenotype is yellow and smooth. Recessive is green and rough. This is another example with peas. So yellow peas and smooth peas or rough peas or green peas. Practice question. And this should not be seen as a practice test question. It's more of a practice question for if you want to try to figure it out, and then I'll explain it, just for instructional purposes. A gamete receives the chromosome containing the Y allele, the dominant Y allele. Use a diagram to explain how the same gamete could receive either an S or a little s. You can pause it and give it a shot, or I'll give you my interpretation of an answer. You could draw many different diagrams. So remember that half of the time we're going to get one of these alleles, half the time we'll get the other. What I've shown here is gamete number 1, gamete number 2, gamete number 3, and gamete number 4. So in meiosis, segregation is going to happen. Segregation of alleles, we'd have them line up at the metaphase plate. This chromosome would be divided. One's going to go left, the other's going to go right. Same thing here. And so 50% chromosomes 1 and 2, they have both gotten the dominant Y. So they're going to both be yellow. And so this is their genotype there. And then the other 50% have gotten the green recessive allele. So let's start with from the beginning again. If a gamete has gotten the dominant Y allele, then they could either get, looking up here again, they could either get the big capital S for smooth, or they could get the lowercase s for rough. Either one. So it could be the dominant one, could be the recessive one. And that is because of random orientation in metaphase 1. Here is a hybrid cross. We're going to be considering two traits, the ones we just talked about, and they're each carried on a separate chromosome. The genes are unlinked, meaning they are not on the same chromosome. This is an example using sweet peas. What is the predicted phenotypic ratio for a cross between two pea plants which are heterozygous at both loci? So heterozygous at both loci. First, you're going to want to reread the question probably and try to figure out how would we write the genotypes of both parents. Here's how. So phenotype was given to you. Well, it's not given to you. They're heterozygous, but if they're heterozygous at both loci, then they're going to be having one of each allele, right? So big S, big Y, little y. And the dominant alleles are going to be expressed. The recessive alleles will be covered up by the dominant alleles. They're both heterozygous. Now we need a Punnett square that is much bigger. We're going to consider each locus or gene one at a time. Let's take one at a time. Let me show you the answers first. Okay, so the gametes are shown in this row and then in this row, possible gametes. Half the time, a gamete will receive a big S. So there's here and here. That's 1, 2 out of 3, 4 times. So that is half the time. Great. And then the other half the time, they will receive a little s. So 3 and 4, that's the other half of the time. So these are where these are gametes showing where the recessive allele has shown up, these with the dominant. Why do we have to show it 2 times? The reason we have to show it 2 times is because there's 2 possibilities. If you get in a gamete a big S, you could get a big Y or a little y. So we have to show these 2 possibilities. Could be both dominant, could be one dominant, the other recessive. The opposite is also true. Or, no, not the opposite, but here's the other half. If a gamete receives a little s and a little s, it could, we need to write it 2 times, little s, and it could receive a big Y or a little y. So 2 options for gametes here. This side here is 100% the same because the parents' genotypes are 100% the same. So the gametes, their possible gametes, will also be the same. Next, what you do is you just draw these down. So if there's a big S here, then you go down this whole column here and add an S there. And you also would add a Y there. Same thing here. You're going to go across this way and just add a big dominant S to every single square or cell in this grid. And I can do the same thing here. Take this big S and bring it all the way across. All those are big S's, at least one big S. I'll just do a couple more. I'm not going to do all of them. So here again, there's a big S there. I'll draw that all the way down. Okay, this big S. I'm going to add a little S to each of these. And basically, you can kind of see how this is going. So if you wanted to know where did these alleles come from, you just look up to here. Well, there's a little s from there, a little s from there, and a little s from there. So 2 little s's. Where did the big Y and the little y come from? Well, look up. One came from this parent in this gamete, and then the little y came from over here from that parent. Okay. Yay. There we go. Phenotypic ratios are as follows, and this one I actually have memorized because it's 9, 3, 3, 1. I have it memorized because really common in all biology courses to ask students, tell me what is the phenotypic ratio in a dihybrid cross where both parents are heterozygous, and it always produces a 9, 3, 3, 1 ratio. 9 where both of the phenotypes are dominant, so it's smooth and yellow, both are the dominant phenotypes. And then 3 and 3 where one phenotype is dominant, the other is recessive. So smooth is dominant, green is recessive, and then the opposite is also true. Rough is recessive and yellow is dominant, and then only one where all the alleles that were deposited in these gametes and then the zygotes all were recessive. Let me restate what all of these cells mean. Those are possible genotypes in offspring. These are possible genotypes in the offspring. So you might say, well, what if I just had one pea? What if this pea plant was really unsuccessful? You know, they should have like 500 offspring, but what if they just had one? And you wanted to know, what are the chances that I'm going to have a baby pea that is smooth and green? Well, the chances are 3 out of 16. Why 16? Well, 9 plus 3 plus 3 plus 1 is 16. You also count up these cells, 1, 2, 3, 4, and you realize there's 16 different possibilities. Even though some of them are the same, they're the result of different of the alleles coming from different parents. All right. This is an example question that I've given to you guys. You do not have to do it. This is more difficult than anything on your exam, but I thought you might just like to give it a shot, because if you can do this one, then you can definitely do exam questions that are about this. The exam wouldn't be that much easier, only a little bit easier. This is from a different class I taught, so if you want, you can try it. not necessary though. Here's your answers. This is the answers. Autosomes and sex chromosomes. So let's remember that the reason I'm reviewing this is we have sex-linked genes. We're going to talk about another kind of linkage. So autosomes are everything but the sex-linked genes. All those are the autosomes, and these are the sex chromosomes. X and Y's are the sex chromosomes. So 22 pairs of autosomes and 1 pair of sex chromosomes. So we generally just say we've got 46, but in fact we kind of have like 22 that are not related to sex, and then we have a sex determination, and then we've got 1 pair that is this sort of unusual one, this tiny little Y and the more normal sized X. So this is autosomal gene linkage, and it has nothing to do with sex linkage. That's why I went over that last slide. Nothing to do with sex linkage. These genes are linked because they are pairs or groups of genes which are inherited together, and they're carried on the same chromosome. So what that means is, if you have 2 genes here that have we've lined up the metaphase plate on, we'll say meiosis 1, and you've gotten this gene here, that has no connection to whether or not you're going to get the A allele or the little a allele on this chromosome. No connection whatsoever, because these 2 could switch place due to random orientation in meiosis. So you could get the big B and the big A, or you could get the big B and the little a. So here we go. One more further explanation. Genes carried on the chromosome, same chromosome. So what if we had this big B, and then we have another gene here that's, say, gene C? Well, basically, if you get gene B, it goes that way during metaphase 1, you're very likely going to get C as well. And that is because they're on the same chromosome, so unless crossing over occurs, you're getting both of them, so one or the other. So sex-linked and autosomal gene linkage, just to remind you, they are not the same thing. Sex-linked disorders are carried only on the non-homologous region of the X chromosome, and all alleles are expressed whether they are dominant or recessive, as there is no alternate allele carried on the Y chromosome. Gene-related disorders, which are sex-linked, include red-green color blindness and then hemophilia, which we'll get to later on. Males are mostly affected. Sometimes females are. It's rare. Autosomal gene linkage, that simply refers to genes or loci that are on the same chromosome. So, for example, locus 1, maybe it's a dominant A and a little a. We're going to say it's right there. It's right there. It codes for a voltage-gated sodium channel, okay, passive transport of some sort. And then locus 2, we're going to say that's involved in programmed cell death, maybe some cancer protection and various alleles on that one. So if you get one of these genes, or rather alleles, so if it was a big A and then a big B, those 2, if this chromosome has these alleles that are both dominant, then if you get this chromosome, you get all of the alleles that are on that chromosome. Okay, so genes at loci 1, 2, and 3, they're all linked by being on chromosome number 3. We can call these a linkage group, and so they'll all be inherited together. Independent assortment does not occur between linked genes. So it's lucky for Mr. Mendel that he was studying unlinked genes, because it would have been a little bit more confusing if, say, all the yellow individuals are always smooth coated as well. Standard notation for gene linkage is as follows. We're going to draw 2 lines, right, like we can see right here. And we say that big A and big B, they are both linked. These alleles are linked to each other, and then the recessive ones are also linked to each other, little a and little b. Now, it is certainly possible that you could have a linkage group such as this one over here, where we have a dominant um, allele for a gene there, and then we've got sorry, having a little bit of technical difficulties here. And then we have a recessive allele there, and then maybe this one's got the recessive for the A, and then maybe the dominant for the B. However, I will be describing alleles that are linked always in the manner of being dominant alleles linked together and recessive alleles linked together, simply because it is much easier to explain and not lose track of what we're talking about. So here, let's go through an example now. In sweet peas, the genes for flower color and pollen grain shape are carried on the same chromosome. So that means that they are linked. Plants which are heterozygous at both loci are test crossed. So we're going to think what ratio of phenotypes is expected. So remember, if it is a test cross, then the test cross, one of the parents, is going to be 100% recessive. So one of the individuals, their phenotype is going to be white and short. So this is the test cross plant, because white and short, those are both the lowercase letters here, those are both recessive traits. So this is all, these chromosomes are all containing recessive alleles for the two genes we're interested in. Here, the plant to be test crossed, we know its genotype because in the question they have said that it is heterozygous. In the past test cross I described, we were looking at a plant where we weren't sure if it was homozygous dominant or heterozygous. In this case, what we're looking at is we're essentially wondering these two genes, we've been told that they are carried on the same chromosome. So we want to test cross them to see, are these two traits going to be inherited in a manner that is consistent with them being linked to each other? So let's take a look. What we can see with the notation here is that one chromosome, this one up here, has both dominant alleles. This chromosome has both recessive alleles. So, but the genotype in the standard format would be written big P, little p. My pen is not working. Big L and little l. Okay, so what we expect to see is as follows. The test cross is going to show up right down here, and we can see that there's all little p, little l. They're all only the recessive alleles are in their gametes. So I've simplified the Punnett square here quite a lot. Possible gametes, there's only one possibility. It's always going to be these recessive alleles, a little p and a little l. And then this individual, the chromosomes will segregate during metaphase 1. So gametes will either receive big P, big L, or little p, little l. So one gamete could be this fellow right here. The other gamete could be this fellow right here. That's what we assume if the genes are linked. That's what we would assume. All right. So that's what we get. We would expect a ratio of 1 to 1, half of them being what we're looking at here, purple and long, big P, big L, little l, and then the other half being all recessive alleles and recessive phenotypes. However, what if this happens? Plants which are heterozygous at both loci are test crossed, but a small number of purple short and white long individuals have appeared in the offspring. Can you explain what has happened? So purple short, let's look at what that is. So purple and short. What that means is an individual has gotten a dominant allele and a recessive, a dominant phenotype and a recessive phenotype. What we just looked at, you can rewind the video a little, is that only plants would only have either all dominant phenotypes or all recessive phenotypes. So what has happened here, some of you have probably guessed. The answer is that these alleles have been recombined. And this is so of alleles, recombination of alleles as a result of crossing over between non-sister chromatids. So crossing over back to haunt us again is the exchange of alleles, and it gives new genotypes and more variation in gametes, new genotypes of gametes. So a small number of purple short, those are the recombinants, and white long, a small number. So it didn't happen in every case. It was a small number, not that many have appeared in the offspring. So what has happened? All right, start at the beginning. Let's take just one parent. The one that was heterozygous. So big L, big P, this is the chromosome that has the dominant alleles. And then here we have little l, little p. This is the chromosome that has all the recessive alleles. In meiosis, or prior to meiosis, we get replication of the chromosomes. So we've seen that there. The possible gametes, genotypes from this individual are either all the dominant alleles or all of the recessive alleles. Over here, coming back, the test cross, remember, that the test cross is homozygous recessive. So the test cross plant only produces gametes that are little p, little l. So this is it. That's all that is going to come from the test cross. So we should have only big P, big L, or little p, little l. So these are the expected. Should be 1-to-1. But crossing over during prophase occurs. So what we can see is this little allele, recessive allele, little p, has been exchanged for a big P. So now what the result is going to be of that is that on one of these sister chromatids, this one, now there's a big L and a little p. So and then this one's also a recombinant. It's got a big P and a little L. So now there's a few gametes that have been produced, because this doesn't always happen. In a round of a cellular division in one human being producing a sex cell, it might happen like a dozen times or so. So it's pretty rare. So we call these gametes recombinants because non-sister chromatids have exchanged alleles. Same genes, but maybe different alleles. So big P, little l, little p, little l. And remember, this is showing one chromosome here, and this one's showing another little chromosome there. Okay, so drawing the Punnett square for this, and again, this I won't ask you to reproduce this level of complexity. This is only for instructional purposes. So gametes produced from non-recombinants are either both dominant or both recessive. Here's the test cross gametes, all homozygous recessive, all little p, little l. The recombinants, small number, because crossing over is fairly rare, is we have now a big E and recessive L and a little p and a dominant L. Mixture of dominant and recessive phenotypes. Crossing over just occurs occasionally. Additionally, it is more likely to happen between linked genes which are further apart. Linked genes which are further apart on the same chromosome. Your book talks a little bit about mapping how far away these genes are using crossing over, and I am not going to be including that on your test. So you're not going to be asked to talk about chromosome mapping or gene mapping for this test. This is just a summary of crossing over from meiosis, and I welcome you to just take a second look at it. Synapsis, chiasma formation, and then recombination if alleles are exchanged. Crossing over, again, just a bit of a review from meiosis. So over here, just took a look at what's going on the left side of the wild type, if they're linked. So big D's and little e's are linked in this case. So if we get division here, this chromosome. Oh, excuse me, what am I talking about here? What we're looking at here is sister chromatids. So the result from this division, this is showing the sister chromatids here. This is where the metaphase plate would be. So gametes would either be big D, little e, or a little d, big E. Recombinants would exchange alleles, big E for little e. So now we produce some recombinants because now these sister chromatids are no longer the same. This sister chromatid, big D, little e, is not the same as this one anymore. Big D, big E. Not the same. So when metaphase 2 occurs and they are separated, we get two different gametes. Big D, little e, and big D, big E. And same thing down there. This I'm not going to go over. It's a little bit more information for you, just mostly about just remembering that genes such as maybe this one up here, this is the name of the real loci, 3p26.1. This one and this one down here, 3q28. These ones are more likely to have a recombination event occur between them because they're farther apart and there's all this big space on the chromosome where crossing could occur, which would separate them. That's the big idea there. What is shown here, and this again is instructional purposes only. You're not meant, this is more difficult than an exam would be. So which description best fits this image? You can pause the video and take a look at it for a moment. Answer is B. 4 chromatids, 2 chiasmata, and 2 centromeres. So the centromeres are here and here, and then the chiasmata formation are there and there, and then of course the 4 sister chromatids are just the 4 lines that have gotten themselves a little tangled up due to chiasma formation. There you go. Gene linkage and recombination. Just a couple of practice questions for you. The gene for kernel color and waxiness are linked in the corn plant, Z maize, in a cross between a plant that is homozygous dominant at both loci with a plant that is heterozygous at both loci. Identify the following genotypes as A, regular, B, recombinants, or C, impossible. You can pause it, and because now I'm going to show you the answers. Those are regular. These ones are the recombinants. And these ones are impossible. Please feel free to post on the discussion forum if you're unclear on that one. 2 genes are linked as shown here, big E and little m, and then also little e and big M. The genes are far apart such that crossing over between the alleles occurs occasionally. Which statement is true of the gametes? You can pause it because I will show you the answer in a moment. D. There will be more big E little m gametes than little e little m gametes. You can also feel free to ask about this one in the discussion forum. Hopefully that's an explanation. This is just about the last topic for this chapter. It is gene interaction. One way to think about it is shown here. The expression of one gene is dependent upon the prior expression of another gene. Here, what we can see is a metabolic pathway where gene A codes for some kind of an enzyme. This enzyme turned this substrate here into something that's, say, a pancake. So now we've converted this substrate to a pink product. Gene B converts the pink product, now we could sort of think of it as a substrate, into the final product, which is purple. So gene B requires gene A to do its work before it can do its work. So first, gene A determines whether color is present. This is kind of similar to albinism in human beings and many other species. The second, gene B, is only expressed, meaning it only matters if A is first expressed. It determines which color will be produced. So we can sort of think as the capital letters as being dominant and the recessive being recessive, except that the B gene, whether it's big B or little b, first we need to look at what's going on with the other gene, the A gene. So these two top bits here, where there's these dashes, let me erase this. I'm starting to worry that my computer is about to crash because it is not functioning correctly. This one right there as well. It's got a dash here. What this means is simply that this is a genotype and phenotype. And what we're meaning here is if there's at least one big A, the second copy doesn't matter, and then two little b's, then it's pink. Then this one up here, if there's at least one big A and at least one big B, then that means it's purple. And this should make sense because essentially, if it's an enzyme and the dominant allele codes for functioning, it's happening. What that means is the dominant allele produces an enzyme. It produces the protein. So as long as there's one, enough of this protein is produced and the reaction is catalyzed. Second one is also true. Gene B, as long as we have one dominant allele, it's going to be producing this enzyme, which will convert the pink substrate product molecule to the final purple product. In looking at the generational inheritance patterns with this one is really wacky. For example, parent phenotypes in gene interactions. Parent phenotype, pure breeding pink crossed with pure breeding white. So what that means is that pure breeding pink and pure breeding white, you really got to think about that one carefully, because what that would mean is that if it's pure breeding pink, then that means that this individual, if it is somehow crossed with another pure breeding pink individual, it will always have pink offspring. Then same thing for the white. If it is pure breeding white, then it is always going to have the same phenotype. So, pure breeding white, offspring will always have the same phenotype. All right, so we cross them. And it would be helpful for you if you were going through this on a test. You want to write out the parent genotype. But F1 generation phenotype, it turns out to be purple flowers. And then the F2 is purple, pink, and white. So this is a much more difficult inheritance pattern to track. But basically, you can look at this. F1, if they're purple, then that means that it's going to be this genotype, at least one big B and one big A. What that also means is these pure breeding white parents in the parent generation, they must have been this phenotype. Little a, and then big B, big B. Pure breeds are always homozygous. Oh, I am no longer able to write correctly. My computer is about to crash, I think. Okay, so just some further explanations down here. This is a word that you need to know from your textbook, epistasis, and it's a common type of gene interaction in which the presence of certain alleles of one locus or gene can prevent or mask the expression of alleles of a different locus and express their own phenotype instead. Last slide, polygenic inheritance. So polygenic inheritance means that there are many genes coding for the trait. Example, in human beings, well, skin color is an obvious one because clearly there's not just, well, you could have the dominant allele or the recessive allele, so there's only two phenotypes possible. Clearly not true. We know of at least 3 genes, and I think we know that there are more, but we don't know as much about them. So AA, BB, CC, each one's either dominant or recessive, and that's assuming that there's only 2 alleles for each gene. I believe there are, in fact, more. This is a simplification. What they're showing here is basically, well, the more dark alleles, more dominant alleles you have, then the more dark the skin pigmentation will be. The more of this other type of allele, then the more light skin pigmentation will be. This is a word you need to know. Continuous variation. That's 2 words. Phenotypes typically show continuous variation. What that means, Let me just give you a couple other examples. You'll explain it pretty clearly. Height, weight, rate of reaction in enzymes. Those are all continuous variation. It's not like you're either tall or either tall or short. You could be many, many variations or intermediate heights between tall and short. In fact, an infinite number of variations or intermediate heights. Example of discontinuous variation would be something like number of peas in a pod. You're always going to have either 4 or 5 or 6. It's not going to be 4.312 peas or 5.786 peas. It's always whole numbers. Live or die is did the pea plant live or die? It's one or the other. It might be barely alive, but it's still alive. Number of heads, 1, 2, 3. That's a strange example, but it's not going to be 1.5. All right, so that is it for inheritance. I've left the last little topic there for you to look at. It's gene-environment interaction, so make sure you take a look at that as well.